5,000 years ago, one Scandinavian community faced three separate waves of Yersinia pestis across 6 generations. 1,000 years before Columbus, a form of tuberculosis linked to seals was already infecting people in Peru. And beneath the Mediterranean, a mother and child carried evidence of human tuberculosis from 9,000 years ago. These discoveries reveal that disease was shaping human lives long before modern medicine, and in some cases long before writing. But the evidence is rarely simple. Bones can preserve the damage left by an infection. Teeth can trap fragments of a pathogen's DNA. Neither can tell us everything about how a disease spread, how many people it killed, or what those people believed was happening to them. So, in this countdown, we will separate what the evidence confirms from what researchers are still debating. Let's begin. Number 10. The Balathol skeleton and a disease older than the written record. In 1994, a joint archaeological team began excavating a low mound outside the village of Balathol in Rajasthan, India. The work was directed by archaeologist V.N. Misra. Across several field seasons, the researchers uncovered a fortified Chalcolithic settlement associated with the Ahar culture. The community stood near the wider cultural and trading world of the Indus civilization, but Balathol itself was not a great city. It was a farming settlement built from stone and mud brick. Inside a stone enclosure, the excavators found the skeleton of a middle-aged man. He had been placed on his left side in a tightly flexed position within layers of burned cow dung ash. His bones carried an unusual pattern of damage. More than a decade later, biological anthropologist Gwen Robbins and her colleagues published a detailed analysis in PLOS One. They documented changes in the bones around the nose and upper jaw, inflammation of the tibia, and damage to the hands and feet. Taken together, those lesions were most consistent with advanced lepromatous leprosy, now known as Hansen's disease. This was a paleopathological diagnosis. In other words, the researchers identified the disease from its pattern in the skeleton. They did not recover the bacterium's ancient DNA from this individual. That distinction matters. Several diseases can damage bone, so the team compared the skeleton with possible alternatives, including tuberculosis, leishmaniasis, treponemal disease, osteomyelitis, and other infections. Their differential diagnosis favored leprosy. Radiocarbon dates from the surrounding archaeological layer placed the burial between roughly 2500 and 2000 BC. When the study appeared in 2009, the Balathol man represented the oldest documented skeletal evidence of leprosy. The finding pushed physical evidence for the disease far earlier than the widely accepted South Asian texts dated to around the 6th century BC. For generations, historians often suggested that leprosy moved west from India during or after the campaigns of Alexander the Great in the 4th century B.C. Balathol did not prove where the disease began, but it showed that a severe form was present in South Asia many centuries before those campaigns. The settlement also complicates a simple urban explanation. Leprosy usually spreads only after prolonged close contact, most likely through respiratory droplets. Crowding, poverty, malnutrition, and weakened immunity can increase the risk. Yet, a disease does not require a metropolis to persist. A connected rural community can also sustain transmission, especially when people remain in close contact over long periods. The original researchers proposed that the disease may have moved through the trading networks linking South Asia, Mesopotamia, and Egypt during the 3rd millennium B.C. Archaeology confirms that those regions were connected. It does not reveal the exact path taken by this infection. More recent discoveries have made the story even broader. Researchers have found possible leprosy cases from late Copper Age Hungary. Ancient genomes also reveal considerable diversity among strains of Mycobacterium leprae in medieval Eurasia. Then, in 2025, scientists recovered 2 roughly 4,000-year-old genomes of Mycobacterium lepromatosis, a second bacterium that can cause Hansen's disease from human remains in northern Chile. And in 2026, researchers reported the first ancient Mycobacterium leprae genomes from eastern Eurasia. Those genomes belong to an ancestral branch, whose distribution is consistent with a possible origin in southern East Asia, although the larger origin story remains unresolved. That means Balathol is no longer a solitary clue to a simple source population. It is one early point in a much larger and more complicated history. What makes the skeleton important is not that it solves the origin of leprosy. It does something more defensible and just as striking. It shows that by about 4,000 years ago, Hansen's disease had already reached a man in a farming community in Rajasthan and had progressed far enough to leave a permanent record in his bones. Number 9. The Chinchorro world and a parasite with a 9,000-year record. Long before ancient Egyptians became famous for preserving the dead people on the Pacific coast of northern Chile, were already practicing deliberate mummification. The Chinchorro were fishing and foraging communities who lived along one of the driest coastlines on Earth. Some naturally preserved bodies from the region date to around 7020 BC. Deliberate Chinchorro mummification began later by about 5050 BC. Their methods could be extraordinarily complex. In some cases, embalmers removed soft tissue, reinforced the skeleton with reeds or sticks, reshaped the body, rebuilt the face with clay, and covered the skin with pigments. These were not the actions of a distant royal court. They were community traditions applied to adults, children, and infants. But the dry coast preserved more than mortuary practice. It also preserved evidence of infection. In 2004, paleopathologist Arthur Aufderheide and an international team published a study of 283 mummified bodies from northern Chile and southern Peru. Most of the bodies had been preserved naturally rather than deliberately mummified. They represented 11 major cultural groups and covered an extraordinary interval from approximately 7050 BC to the period of the Spanish conquest. The researchers tested tissue samples for DNA from Trypanosoma cruzi, the parasite that causes Chagas disease. About 41% of the tested tissue extracts produced a positive result. The prevalence showed no statistically significant difference between the cultural groups in the sample. It also showed no clear difference based on age or sex. That does not mean 2 out of every 5 people along the entire ancient coast died from Chagas disease. A positive result shows infection, not necessarily the cause of death. Many people infected with Trypanosoma cruzi remain without symptoms for years. Others later develop serious heart or digestive complications. Even with that caution, the continuity is remarkable. The parasite was present when the earliest sampled Chinchorro communities lived on the coast. It was still present thousands of years later among societies with different technologies, economies, and ways of life. How did it persist for so long? Today, Trypanosoma cruzi is often transmitted by triatomine insects commonly called kissing bugs. The insects can live in cracks, in houses, and animal shelters. But the parasite also circulates in wild mammals, and humans can acquire it through several routes, including contaminated food and congenital transmission. The original study did not conclude that one style of adobe and thatch housing remained unchanged for 9,000 years. Instead, the researchers argued that the parasite cycle among wild animals was probably already established before the first humans settled that part of the coast. When people arrived, they became new hosts inside an older ecological system. Housing and settlement patterns may later have intensified exposure in some communities. But they were not the only possible route, and the archaeological sample cannot reconstruct one unchanging domestic lifestyle across the entire 9,000-year span. The region carried another health burden as well. Research led by Bernardo Ariaza and other scholars has documented elevated arsenic in local water sources. Chronic arsenic exposure can damage health and may have contributed to pregnancy loss and infant mortality in some communities. That has led researchers to ask whether unusually frequent death, especially the deaths of infants and children, helped encourage the development of elaborate mummification. It is an intriguing hypothesis, not a proven explanation. We cannot say that Chagas disease or arsenic caused the Chinchorro to create the world's earliest known tradition of artificial mummification. Mortuary customs grow from belief, memory, identity, and social relationships that bones alone cannot recover. What the evidence does show is powerful enough. Across 9,000 years of human remains from the coastal Andes, the same parasite appears again and again. Cultures changed, burial customs changed. The relationship between people, animals, insects, and the desert environment kept the infection alive. Number 8. Atlit Yam, the drowned village, and tuberculosis in a mother and child. In 1984, marine archaeologist Ehud Galili was surveying the seabed off Israel's Carmel coast when he recognized the remains of a submerged settlement. The site became known as Atlit Yam. Today, it lies roughly 8 to 12 meters below the Mediterranean. When people lived there about 9,000 years ago, it stood on dry land beside freshwater sources. The settlement covered approximately 40,000 square meters. Archaeologists found stone-built houses, courtyards installations for food production, and a stone-lined well sunk more than 5 meters into the ground. They also found 7 upright stones arranged in a semicircle near a freshwater spring. Some of the stones carried cup-like depressions. The arrangement may have had ritual significance, although its precise purpose remains unknown. Among the human burials were the remains of a woman and an infant placed together. Both skeletons showed lesions compatible with tuberculosis. In 2008, a team led by Israel Hershkovitz and Helen Donoghue published a molecular analysis of the remains. They used several independent forms of evidence. First, they amplified DNA from 5 genetic regions associated with the Mycobacterium tuberculosis complex. Then they used high-performance liquid chromatography to detect mycolic acid's distinctive lipid biomarkers from the bacterium's cell wall. The results supported tuberculosis in both individuals. The infant may have acquired the infection from the woman, possibly through close contact after birth. The original researchers also discussed neonatal transmission, but the exact relationship between the two individuals and the route of infection cannot be established with certainty from the burial alone. At the time, this finding pushed molecularly supported evidence of human tuberculosis, back by roughly 3,000 years. It also challenged an older story about where tuberculosis came from. For many years, a common explanation held that human tuberculosis originated when Mycobacterium bovis jumped from domesticated cattle into people after animal husbandry became established. The Atlet yam evidence did not fit that model. The detected genetic signatures were consistent with lineages of the Mycobacterium tuberculosis complex known in humans. They did not support cattle as the source of infection in this case. Atlet yam already had agriculture and domesticated animals. The researchers suggested that living with animals may have supported a larger and denser population, indirectly creating better conditions for human-to-human transmission. But the animals themselves were not identified as the source. The larger evolutionary history of tuberculosis remains complex. Molecular clock estimates have changed as scientists have added ancient genomes and improved their methods. What Atlet yam provides is a secure archaeological demonstration that tuberculosis was affecting a settled human community in the eastern Mediterranean roughly 9,000 years ago. The sea adds another layer to the discovery. Atlet yam was not submerged in a single dramatic moment that archaeologists can confidently identify as a tsunami disaster. After the last ice age, global sea levels rose. Local coastal processes also changed the shoreline. Over time, the village was abandoned, and the water advanced across the land. The freshwater well filled with seawater. Houses disappeared beneath marine sediment. The standing stones ended up on the seabed. We do not know how the community understood tuberculosis. There is no evidence that they practiced quarantine, and no surviving account of treatments or beliefs. Those possibilities belong to imagination, not archaeology. What survives is more limited but more concrete. A woman and an infant were buried together. Their bones and the molecules preserved inside them show that human tuberculosis had already become part of village life thousands of years before the first written medical texts. Number 7. Saddlehoyuk and the health costs of early crowded life. For more than 1,000 years, people built, rebuilt, and lived at Satalhoyuk in what is now south-central Turkey. The settlement was occupied from approximately 7,100 to 5,950 BC. At its peak during the centuries around 6,700 to 6,500 BC, researchers estimate that between 3,500 and 8,000 people may have lived there. The houses were packed tightly together across a settlement covering about 13 hectares. There were no streets between most buildings. Residents moved across the roofs and entered their homes by ladder through openings above. Inside walls and floors were repeatedly covered with fresh layers of plaster. The dead were often buried beneath the floors. Çatalhöyük is sometimes called the world's first city. That description is catchy but debated. It had a large, dense population and many features of proto-urban life. Yet it lacked the streets, public buildings, centralized government, and clear administrative hierarchy associated with many later cities. Calling it one of the earliest large proto-urban settlements is more accurate. In 2019, a team led by bioarchaeologist Clark Spencer Larson published a major study of the human remains excavated at the site. The research drew on 25 years of work and followed biological changes across the settlement's long history. The bones revealed several costs of permanent crowded living. About 10 to 13% of adult teeth had cavities. The community's grain-heavy diet, rich in wheat, barley, and rye, exposed teeth to fermentable carbohydrates more often than many earlier hunter-gatherer diets had. Up to one-third of the examined individuals from the early phase showed evidence of infection on their bones. This was not a diagnosis of one named epidemic. Skeletal inflammation can have many causes, and most infections never leave a clear mark on bone. The evidence instead points to a substantial burden of disease in the community. Crowding probably contributed. So did sanitation. Animal and human fecal material has been identified in and around houses. Trash areas and animal pens stood close to domestic spaces. Sheep and goats were central to the economy, and repeated contact between people, animals, waste, and contaminated surfaces created many opportunities for infection. The skeletons also preserved evidence of interpersonal violence. In a sample of 93 skulls, 25 people had healed fractures caused by blows to the head. 12 had been injured more than once. The shape of the wounds suggested hard, rounded objects, and clay balls of suitable size have been found at the site. 13 identified victims were female and 10 were male. Many wounds occurred on the top or back of the skull. The frequency of cranial injury increased during the Middle Period, when the settlement reached its greatest density. That pattern is consistent with rising conflict under crowded conditions, but it does not prove that population density alone caused the violence. Social tensions, household disputes, ritual behavior, and other factors may also have played roles that the bones cannot separate. The burials beneath the houses produced another surprise. Researchers compared inherited features in the teeth of individuals buried under the same buildings. Many were not close biological relatives. This does not prove that they were unrelated in every social sense, or that they consciously rejected blood-based family. It does show that burial groups were not simply nuclear families organized only by biological kinship. Membership in a household may have depended on residence, work, ancestry understood through a different line, adoption, ritual affiliation, or relationships that have no direct modern equivalent. The evidence leaves the question open. Over time, the environmental demands of the settlement also increased. Residents in the later period walked farther than earlier inhabitants according to changes in the cross-sectional shape of their leg bones. Farming, grazing, and gathering firewood may have required longer journeys as nearby resources were depleted and the regional climate became drier. Çatalhöyük did not invent disease, nor does the site prove that every city must produce the same health crisis. What it captures is a transition. People gained the stability and productivity of settled farming. At the same time, dense housing, grain-based diets, accumulating waste, close contact with animals, environmental pressure, and human conflict created new combinations of risk. The problems feel modern because the underlying biology has not changed. Put many people together for generations, and the organization of food, water waste, animals, and personal space becomes a matter of survival. Çatalhöyük records one of humanity's earliest large-scale attempts to negotiate that balance. Number 6. The tuberculosis that crossed an ocean inside seals. Along the arid coast of southern Peru, archaeologists recovered skeletons of people who lived around 700 to 1000 AD, centuries before Europeans reached the region. Some of those skeletons showed damage consistent with tuberculosis. The most familiar signs included collapsed and fused vertebrae deformation of the spine and lesions on the inner surfaces of the ribs. Those changes can suggest chronic disease, but skeletal appearance alone cannot identify every case with certainty. Researchers screened remains from pre-contact South America and identified 68 individuals with lesions compatible with tuberculosis. Recovering readable pathogen DNA was far more difficult. In 2014, a team led by Kirsten Boz reconstructed 3 complete ancient genomes from the Mycobacterium tuberculosis complex. The remains came from people associated with coastal communities in Peru and were approximately 1,000 years old. The result was unexpected. The ancient strains were not closest to the human-adapted tuberculosis lineages circulating around the world today. They clustered with Mycobacterium pinnipedii, the lineage associated with pinnipeds, seals, and sea lions. The most likely route began in Africa. An ancestor of the tuberculosis complex probably infected marine mammals on the African coast. Seals or sea lions then carried the pathogen across the South Atlantic and through southern hemisphere waters. Thousands of years later, infected animals or their tissues brought the bacterium into contact with people on the Pacific coast of South America. This was not a direct Pacific crossing from the Old World to Peru. The proposed movement was from Africa through the Atlantic and around the southern marine range before reaching the Pacific coast. The bacterium then crossed the species barrier again from pinnipeds into humans. Coastal Andean people hunted marine mammals, processed their carcasses, ate their meat, and used their hides and other tissues. Any of those activities could have created exposure. Images of seals and sea lion hunting also appear in Andean art, showing that the animals had economic and cultural importance. But the exact moment of transmission is unknown. It may have occurred during butchering through food, through respiratory exposure, or through repeated contact with infected tissue. Archaeology cannot identify a particular ceremony or prove that a sacred relationship caused the infection. Later research made the pattern even more interesting. In 2022, scientists reported additional ancient Mycobacterium pinnipedii genomes from human remains in Colombia and Peru. Some infected people lived far inland, where direct contact with live marine mammals would have been unlikely. That suggests the pathogen-infected animal products or infected humans moved through exchange networks after one or more animal-to-human spillovers. It was not simply a series of isolated infections on a beach. The discovery changed the history of tuberculosis in the Americas. Before European contact, people in South America were already living with members of the Mycobacterium tuberculosis complex. The strains identified in these particular individuals came from a lineage adapted to marine mammals. After Europeans arrived, Old World human-adapted tuberculosis lineages largely replaced the ancient pinniped-related forms. As a result, the earlier disease history became almost invisible until ancient DNA recovered it. This does not mean pre-contact Peru experienced one continuous tuberculosis epidemic. 3 genomes cannot establish the prevalence of infection across a continent, and the many skeletons with compatible lesions did not all yield pathogen DNA. What those genomes establish is narrower and extraordinary. A mammalian pathogen with roots in Africa entered marine mammals, traveled across oceanic ranges, reached South America, and caused tuberculosis in humans before European colonization. The ocean was not a barrier. It was part of the transmission route. Number 5. The hepatitis B lineages that vanished from the human record. Hepatitis B remains one of the world's major viral infections. It can become chronic damage to the liver and lead to cirrhosis or cancer. For a long time, scientists knew remarkably little about its deep history. Viruses usually leave no recognizable marks on skeletons. Their genetic material is fragile. Unless viral DNA survives inside a tooth or bone, an ancient infection can disappear without a trace. That changed when researchers began screening large ancient human DNA datasets for fragments that did not belong to the human genome. In 2018, a team led by Barbara Muleman and Terry Jones examined ancient genomic data from Western and Central Eurasia. They detected hepatitis B virus sequences in 25 individuals. 12 infections preserved enough material for the researchers to reconstruct full or partial viral genomes. Those genomes ranged from about 800 to 4,500 years old. At the time, the oldest example placed genetically confirmed hepatitis B in the Bronze Age. The ancient viral family tree did not match the modern map. Some ancient strains fell near lineages that still exist. Others belonged to branches with no known living descendants. Viral forms that once circulated across parts of Eurasia had apparently declined or disappeared. That was not the end of the story. A separate study in 2018 recovered complete hepatitis B genomes from Neolithic individuals approximately 7,000 and 5,000 years old. Then, in 2021, a much larger study generated hepatitis B genomic data from 137 ancient people across Eurasia and the Americas. The individuals lived between roughly 10,500 and 400 years ago. The researchers estimated that the most recent common ancestor of known hepatitis B lineages lived between approximately 20,000 and 12,000 years ago. They also found the virus in European and South American hunter-gatherers during the early Holocene. So, hepatitis B did not begin in the Bronze Age. The Bronze Age study was an early window into a history now known to extend much farther back. The larger dataset revealed repeated population scale changes. After farming spread into Europe, hepatitis B lineages associated with Mesolithic hunter-gatherers were largely replaced by a lineage probably dispersed with early farmers. That lineage persisted across Western Eurasia for roughly 4,000 years, then declined around the end of the second millennium BC. Only a rare modern form genotype G appears to preserve part of that prehistoric diversity. This does not mean ancient strains vanished because they suddenly became more dangerous. A viral lineage can decline when its host population changes, when competing lineages arrive, when transmission networks shift, or through demographic chance. Nor does the incomplete ancient record undermine modern vaccines or treatment. Today's hepatitis B vaccine targets a surface antigen shared across known human genotypes and remains highly effective. Clinical treatment is based on observed viral biology, immune response, and evidence from living patients, not on the assumption that every ancient lineage has been recovered. Ancient genomes contribute something different. They reveal the range of evolutionary change that occurred over thousands of years. They show that modern geographic distributions are temporary outcomes, not permanent features of the virus. Most importantly, they expose a form of survivorship bias. The hepatitis B family tree we see today contains only the branches that reach the present. Ancient DNA has begun to restore some of the missing branches, and every new sample makes the history less linear than it appeared before. Number 4. The extinct smallpox lineage found in Viking Age teeth. For decades, one of the most famous possible cases of ancient smallpox was the mummy of the Egyptian pharaoh Ramses V, who died in 1157 BC. The skin appears to carry pock-like marks, but appearance alone cannot confirm smallpox, and researchers have debated the diagnosis. No complete variola genome has been recovered from the pharaoh. Genetic evidence came from a very different setting. In 2020, an international team led by Barbara Meuleman published the discovery of variola virus DNA in 13 individuals from Northern Europe. 11 of them dated from approximately 600 to 1050 AD, overlapping the Viking Age. 4 carried enough viral DNA for the researchers to reconstruct high-coverage or near-complete genomes. The team had screened material from 1,867 ancient individuals. However, the relevant Viking Age subset was much smaller. The 11 Viking Age detections came from 525 individuals dated to that period. That distinction is important. The sample was assembled from archaeological remains, preserved well enough for testing. It was not a random survey of the Viking Age population, so the positive results cannot be converted directly into a general infection rate. Even so, the genomes transformed the smallpox family tree. The Viking Age viruses belonged to a previously unknown branch of variola. That branch was not the direct ancestor of the smallpox viruses circulating just before eradication. It was a sister lineage that later disappeared. The ancient viruses also carried different patterns of active and inactive genes. Some researchers have suggested that these changes could have influenced how the disease behaved. But ancient genomes alone do not tell us whether the Viking Age illness was milder, how often it caused the classic rash, or how many infected people died. Finding viral DNA in a tooth shows that the virus reached the bloodstream. It does not prove that smallpox caused that individual's death. The geographic spread was still impressive. Positive individuals came from several locations across Northern Europe, with additional genetic evidence connected to Western Russia. The distribution demonstrates that this extinct lineage circulated across a broad region during the Viking Age. Viking communities were highly mobile. Their ships linked Scandinavia with the British Isles, the North Atlantic, the Baltic, and river routes extending deep into Eastern Europe. Mobility could have helped a respiratory virus travel. Close living quarters in houses and ships could also have supported transmission. But the genomes cannot place infected sailors on specific voyages. They do not prove that this variola lineage reached every territory visited by Vikings, and they do not identify one continuous outbreak. The historical setting needs care as well. This was not a world without writing. Runes existed in Scandinavia, and neighboring societies produced chronicles and annals. What is missing is a surviving written account that can be securely matched to this particular viral lineage. People could describe fever, rash death, and community suffering without knowing what a virus was. A written description, even if one survives, may not be medically specific enough to identify variola centuries later. The lineage eventually went extinct, Scientists do not yet know exactly when or why. It may have been replaced by other variola lineages. It may have declined as immunity and transmission networks changed. Or it may have disappeared through a demographic process that left no clear historical marker. Smallpox itself was eradicated in 1980 after a global vaccination campaign. The Viking Age genomes do not change that achievement. They reveal that the virus once had a more diverse evolutionary history than its final surviving lineage suggested. 13 people preserved the evidence. Without DNA in their teeth, an entire branch of one of history's most consequential viruses would remain unknown. Number 3. 3 waves of Yersinia pestis across 6 generations. Around 5,000 years ago, farming communities across parts of northern Europe entered a period of demographic decline. The number of radiocarbon-dated human remains and archaeological sites fell in several regions. The causes remain debated. Agricultural crisis, environmental pressure, social conflict, migration, and infectious disease have all been proposed. At Frelsegården in the Falbygden region of western Sweden, a passage grave preserved an unusually detailed view of one community living near the beginning of that decline. Archaeologists excavated the tomb in 2001. It contained the mixed remains of dozens of people who had been buried over time. In 2019, a team led by Nicolas Raskoven reported an early genome of Yersinia pestis from a young woman at the site. The strain belonged to a deeply branching lineage near the base of the known plague family tree. Then, a larger study changed the scale of the story. In 2024, Frederick Valur Searsholm Martin Sikora and their colleagues analyzed ancient DNA from 108 Neolithic individuals. The remains came from 8 megalithic graves and 1 stone cyst in Sweden and Denmark. At least 18 individuals, about 17% of the sampled population, carried Yersinia pestis DNA. The researchers reconstructed 4 multi-generational pedigrees. The largest centered on Frelsegården and included 38 sampled individuals plus 23 relatives inferred from genetic relationships. That produced a family network of 61 people spanning 6 generations. Within the community, the team identified 3 distinct infection events over roughly 120 years. 3 waves, 6 generations. It is important not to confuse the 6 generations with 6 outbreaks. The evidence supports 3 genetically distinguishable infection events across a pedigree that lasted 6 generations. The strains changed between those events. They retained ancestral genetic material, also found in Yersinia, pseudotuberculosis, the closest known relative of Yersinia pestis. The youngest identified strain had lost some of that ancestral material, suggesting continued adaptation. The social structure was just as revealing. The largest pedigree followed male lines. Most women with children appeared to have come from outside the lineage. In one direct example, 2 brothers were buried together at one site while their sister was buried about 8 kilometers away, where she had children and grandchildren. This supports female exogamy, the movement of women between nearby kin groups as part of the community's social organization. It does not prove that incoming women repeatedly reintroduced Yersinia pestis. The study showed movement and infection in the same regional society, but it did not identify marriage networks as the root responsible for each wave. The most difficult question is how dangerous these early strains were. The 2024 study argued that repeated Yersinia pestis infections could have produced excess mortality and may have contributed to the Neolithic decline. The bacterium was detected in a substantial part of the sampled community, and several infection events occurred across a relatively short period. But detecting bacterial DNA is not automatically proof that each person died of plague. These ancient strains lacked some of the adaptations that later allowed efficient, sustained transmission through fleas. Researchers continue to debate whether they caused severe pneumonic or septicemic disease spread by another vector, moved through contaminated food, or produced a clinical pattern different from later bubonic plague. In 2026, a published scientific commentary argued that the Scandinavian infections may have behaved more like foodborne enteric disease, with some cases progressing to dangerous bloodstream infection. Other researchers continue to defend the possibility of highly consequential outbreaks. That debate should be part of the story, not hidden from it. The secure finding is that Yersinia pestis infected people in the same regional community during 3 distinct episodes over about 120 years. Whether it caused the wider population decline by itself is not established. It may have acted alongside harvest failure, malnutrition, violence, and other pressures. For the people buried at Frelsegården, however, the pathogen was not a single distant event. It returned during the documented history of one extended family. Grandparents, children, and later descendants encountered genetically different forms of the same bacterial lineage. Ancient DNA lets us follow those encounters at the scale of individual relationships rather than as an anonymous population curve. Number 2. The plague strains that moved through a changing Europe. In 2015, researchers led by Simon Rasmussen searched ancient human DNA for traces of Yersinia pestis. They screened 89 billion DNA sequence reads from the teeth of 101 people who lived across Eurasia during the late Neolithic and Bronze Age. 7 individuals carried the bacterium. They lived between roughly 5,000 and 2,800 years ago. Their remains came from regions stretching from the Baltic and Central Europe to the Altai and the Eurasian steppe. The discovery placed Yersinia pestis in humans about 3,000 years earlier than the first historically documented plague pandemic. It also revealed that the bacterium changed while major human migrations were transforming Eurasia. Some infected individuals were associated with the Afanasievo, corded ware, Sintashta Andronovo, and Unyedisi archaeological cultures. These groups were not one population, and they did not all move in the same direction at the same time. But several were connected directly or indirectly to the enormous expansion of steppe-related ancestry during the 3rd millennium BC. Ancient human genomes show that pastoralist populations with ancestry related to the Pontic-Caspian steppe moved into Europe. In some regions, especially parts of Central and Northern Europe, they contributed a large share of the ancestry of later populations. That was a major demographic transformation. It was not a complete replacement of every person across the continent. Modern Europeans retain different mixtures of ancestry from Mesolithic hunter-gatherers, Neolithic farmers, steppe pastoralists, and later populations. The proportions vary greatly by region. Additional research recovered more early Yersinia pestis genomes from Europe and Asia. Together, the genomes show that closely related forms of the bacterium were widely dispersed during a period of intense mobility and population change. Did disease help clear the way for steppe migration? Some researchers have proposed exactly that. In this model, early Yersinia pestis spread among established Neolithic farming communities, contributed to population decline and weakened societies before or during the arrival of steppe-related groups. The timing makes the hypothesis plausible. It does not prove causation. The infected individuals belong to different cultural contexts, including populations connected to the migrations themselves. The presence of the pathogen in their teeth cannot tell us whether they were migrants, members of older farming populations, or people descended from both in every case. Nor can 7 positive individuals measure the mortality of an entire continent. The transmission route is another unresolved problem. Many early strains lacked the YMT gene. In later Yersinia pestis, that gene helps the bacterium survive inside the flea digestive tract and supports the classic blocked flea mechanism associated with bubonic plague. Lacking YMT does not mean the bacterium could not survive in a rat's bloodstream. That distinction matters. It means those strains were not equipped for the same efficient long-term colonization of fleas used by later plague lineages. Other routes may have included respiratory droplets, contact with infected tissue, early-phase flea transmission, lice, or contaminated food. Researchers have not reached a single answer. By about 3,800 years ago, at least one known Yersinia pestis lineage in the Samara region carried the adaptations associated with efficient flea-borne bubonic transmission. Before that point, the bacterium's ecology may have been substantially different. The fairest conclusion is also the most interesting. Yersinia pestis was moving across Eurasia, while human populations, economies, and ancestry were being reorganized on a continental scale. Disease may have contributed to those changes. It cannot yet be treated as the single force that caused them. Human migration moved pathogens. Pathogens could alter the survival of human communities. The Bronze Age evidence captures both processes unfolding together without giving us a simple way to separate cause from coincidence. Number 1. The pathogen exchange that may have shaped the meeting between humans and Neanderthals. When Homo sapiens expanded from Africa into Eurasia, they did not enter an empty continent. Neanderthals had lived across parts of Europe and Western Asia for hundreds of thousands of years. Modern humans and Neanderthals overlapped, met, and interbred. Every sustained contact between previously separated populations creates another possibility: the exchange of pathogens. In 2016, biological anthropologists Charlotte Holdcroft and Simon Underdown published a review arguing that the first major epidemiological transition began far earlier than agriculture. The traditional first epidemiological transition describes the rise of settled farming, larger populations, close contact with domesticated animals, and the growing burden of infectious disease. That framework remains useful, but it never meant that hunter-gatherers carried no infections. Humans and earlier hominins lived with parasites, bacteria, and viruses long before anyone planted crops. Some pathogens can persist in small populations through chronic infection, animal reservoirs, repeated reactivation, or movement between connected groups. Hoodcroft and Underdown reviewed genetic estimates for pathogens including tapeworms, tuberculosis-related bacteria, Helicobacter pylori, and herpes viruses. Some estimates placed important splits in Helicobacter pylori populations between approximately 88,000 and 116,000 years ago in Africa, with lineages reaching Europe after about 52,000 years ago. Those dates should not be described as the exact moment the bacterium first infected a human ancestor. Molecular dates estimate divergence and movement in surviving genetic lineages. They come with uncertainty and can change as new samples and models become available. Herpes simplex virus type 2 has an even deeper story. Phylogenetic research suggests that its ancestor crossed from the chimpanzee lineage into an ancestral hominin roughly 1.6 million years ago, probably through an intermediate hominin host. The precise species and route remain hypothetical. The broad lesson is clear: mobile human groups carried ancient infections with them. When modern humans met, Neanderthals' pathogen transfer could have occurred in both directions. Hoodcroft and Underdown proposed that Neanderthals with small and fragmented populations may have been especially vulnerable to unfamiliar pathogens brought by modern humans from Africa. Modern humans could likewise have encountered Eurasian infections carried by Neanderthals. That idea is plausible. It is not direct evidence that disease caused Neanderthal extinction. In 2019, Gilly Greenbaum and colleagues built a mathematical model of the long contact zone between the two populations. Their model showed that reciprocal disease transmission could help maintain a boundary for thousands of years. As limited interbreeding transferred immune-related genes between groups, both populations could gradually gain resistance. Continued migration of modern humans into Eurasia could then destabilize the contact zone and allow expansion into Neanderthal territory. This was a model demonstrating a possible mechanism. It did not identify a specific prehistoric epidemic, recover a pathogen from a Neanderthal, or prove that disease determined the final outcome. Neanderthals disappeared as a distinct population by about 40,000 years ago. Researchers continue to examine many interacting explanations: small population size, inbreeding, climate instability, competition, differences in social networks, repeated migration by Homo sapiens, assimilation through interbreeding, and possibly disease. There was probably no single cause. The counterfactual is still worth considering as long as it is labeled clearly. If Neanderthal-adapted pathogens had affected incoming modern humans more severely, early Homo sapiens populations in Eurasia might have expanded more slowly, or failed in particular regions. But science cannot calculate how close history came to a complete reversal, and the claim that the margin was small should not be presented as fact. What genetics can demonstrate is that the encounter was not simply a replacement. Modern humans and Neanderthals had children together. Most present-day populations outside Africa carry roughly 1 to 2% Neanderthal ancestry, although the amount varies. Research has also detected smaller Neanderthal-derived segments in African populations, largely introduced through later migrations back into Africa. Some apparent matches also reflect even earlier gene flow from modern human ancestors into Neanderthals. Neanderthal-derived sequences have therefore been detected in populations both inside and outside Africa, although their amounts and histories differ sharply. Some inherited Neanderthal variants affect immune function. Others influence skin metabolism and susceptibility to particular diseases. Their effects can be helpful in one environment and harmful in another. Those variants are genetic evidence of contact. They do not identify the infections exchanged during that contact, or reveal which population suffered more from them. This final case is therefore different from the others in the countdown. At Balathol disease, left lesions in a skeleton. At Atlit Yam, researchers recovered bacterial DNA and lipids. In Viking Age teeth, they reconstructed viral genomes. In the Neanderthal story, the direct pathogen evidence is still missing. What we have is an evolutionary argument supported by pathogen histories, human genetics, demographic vulnerability, and mathematical modeling. It is not a solved case. It is a testable framework for understanding one force that may have operated whenever two long-separated human populations came into contact. That distinction makes the story more credible, not less compelling. Bones and teeth can reveal infections that no written record preserved. Ancient DNA can recover branches of viruses and bacteria that vanished thousands of years ago. But the most responsible conclusion is often not. Now we know exactly what happened. Now we know which explanations the evidence can support and which questions remain open. If you want to see more videos like this, click the video on screen now and make sure to subscribe.